Hi guys, time for some respiration. We're going to be doing aerobic respiration and anaerobic respiration in eukaryotic organisms, which means they have a mitochondria, which is a organelle specialized for respiration. Okay, aerobic respiration in general. Okay, don't forget prokaryotes can also respire aerobically and anaerobically. They use their mesosome instead of a mitochondria, which is kind of like foldings of the cell membrane. Okay, so respiration's split up into a few steps. Glycolysis, the link reaction, which is quite small, the Krebs cycle, and oxidative phosphorylation. I've gone into some extra detail over here about Krebs and oxidative phosphorylation, which we'll get to in a minute. Okay, so first stage, glycolysis occurs in the cytoplasm. In comes your one glucose molecule from your food, from your bloodstream, tissue fluid, or maybe from the chloroplast if this was a plant cell. We add in two ATP molecules. They donate a phosphate group each, which is dephosphorylation, to turn glucose into hexose bisphosphate. So basically glucose with a couple of extra phosphate groups stuck on. Then it's split in two and two hydrogen molecules ping off, and it's split into two triose phosphate. Triose means three, and it's now two 3-carbon sugars. The two hydrogens that ping off are picked up by a coenzyme called NADH. Okay, so it becomes reduced NADH and goes off towards oxidative phosphorylation. More about that later. Then it's phosphorylated, so dephosphorylated. ATP is phosphorylated to make 4 ATP. So we're getting a net gain of 2 ATP molecules in glycolysis. 2 ATP molecules already, that's fab. Substrate level phosphorylation is the name of the way that this ATP has been made, okay? Substrate level. That means it's being made not through oxidative phosphorylation, okay? And don't forget, it's all about making ATP. So we've got 2 ATP molecules already. Fantastic. Pyruvate is sort of the end molecule of glycolysis. So we've got 2 of those. They're 3 carbons each. Okay, these are then picked up by the link reaction. The link reaction involves moving pyruvate into the matrix of the mitochondria. In that process, though, it's converted to an acetyl group, which means removing some carbon. Okay, so we remove 1 carbon from each pyruvate molecule, and that's lost as carbon dioxide. We also remove another couple of hydrogen molecules taken away by the coenzyme NADH. And then another coenzyme A, picks up the acetyl group and becomes acetyl coenzyme A. Remember, there's 2 of those because there were 2 pyruvate molecules. Okay, so all we've done really is moved a carbon off and moved it into the mitochondria, into the matrix. Okay. Then it enters the Krebs cycle. The Krebs cycle is a little bit more complicated than that, but basically we lose more carbon dioxide. We also make a couple more molecules of ATP by substrate level phosphorylation, and lots of reduced coenzymes. NADH again, there's 6 of those made, and reduced FAD as well, 2 of those are made. All of these carrying their hydrogen off to the oxidative phosphorylation reactions. Oxidative phosphorylation is pretty much needs a lot of hydrogen, so that's why they're taking all the hydrogen there. So we've got a little bit of waste products already. We've got quite a lot of carbon dioxide being produced as waste products, which, you know, when you're breathing out is a waste product, carbon dioxide. This is where we get it all from. Let's look at Krebs in a bit more detail. So the Krebs cycle, here comes acetyl coenzyme A, bringing in acetyl group. It's picked up by oxaloacetate, which is a 4-carbon molecule, to make citrate, which is a 6-carbon molecule. We then lose a carbon by carbon dioxide, lose another hydrogen to make some reduced NAD. You don't need to know the name of this intermediate here, but it is a 5-carbon molecule. Another loss of carbon. When we lose carbon, it's called decarboxylation. Clues in the name. Another ATP molecule made here by substrate level phosphorylation. 2 NADH made, reduced there, and there's the FAD. So this is per 1 glucose molecule. Okay. Right, so a lot of hydrogen on its way over to oxidative phosphorylation. This is a two-step process, actually, oxidative phosphorylation. It's divided up into the electron transport chain, which is this bit here, and then chemiosmosis, which is the actual production of ATP using ATP synthase. Okay, so what happens, I'll go into it in a bit more in a second, is lots of hydrogen ions build up in the intermembrane space, and then they diffuse down a diffusion gradient through ATP synthase, which is this little purple molecule there on the cell on the membrane. And as they diffuse along through ATP synthase, it helps sort of turn this little weird ATP synthase molecule, and as it turns, it generates ATP. It's quite clever. You can make about 2.5 molecules of ATP for one reduced NADH coenzyme, and about 1.5 for FAD. Older textbooks will say 3 and 2, so watch out for that. We're only just learning about how this really works. So in more detail then, here we go, oxidative phosphorylation. So this is the matrix, this is the intermembrane space, so in here, in between the cristae and the outer membrane. So here comes our reduced NAD and reduced FAD. They donate their hydrogen to the first and second proteins in the electron transport chain. And the hydrogen is split into electrons, which are going to be this blue line, and hydrogen protons or hydrogen ions. Okay, the electrons have a lot of energy. They're high energy. Well, they're not molecules, are they? They're subatomic particles, so they're really high energy. They get passed from protein to protein. So FAD comes in here to protein number 2, and NAD to number 1. And as their electrons move along the transport chain, they have enough energy to move the protons into the intermembrane space. Up they go. So we end up with a really high concentration of hydrogen ions up here in the intermembrane space, which then down a diffusion gradient diffuse through ATP synthase, and that makes ATP. So at the end we're left with lots of hydrogen and lots of electrons. So what we do is connect it both to oxygen. That's why oxygen is called the final electron acceptor, to make lots of water. So that's another waste product of respiration. So this whole process is going on all over the cristae, all over these inner mitochondrial membranes, which is why the cristae are enfolded to increase surface area for lots of ATP synthase. And you can see along here, look, there's the next electron transport chain right next to it. So it's going on all over the cristae, making lots and lots of ATP for every glucose molecule. Okay, whoops, sorry. Right, something else I want to talk to you about is the respiratory quotient. This is a calculation you need to know, and it shows you sort of how much energy is released relatively for each molecule. Okay, so respiratory quotient for carbohydrates is 1, lipids is 0.7, and proteins is about 0.8. For anaerobic respiration, because obviously no oxygen is used, it's infinity. You also need to be aware of the energy they produce. Obviously you know that lipids produce way more energy. This is kilojoules per gram. Okay, so you might be asked to calculate the RQ, but if you already know them, it'd be a lot easier just to recall them. Okay, anaerobic respiration. Anaerobic respiration is without oxygen. There's no final electron acceptor, as in oxygen. So the electron transport chain stops, so the Krebs cycle stops, so everything stops. In mammals, it's called lactate fermentation, because you end up with lactate. And in yeast and plants, it's called ethanol fermentation, because you end up with ethanol. Some organisms just like working anaerobically. They've evolved for it, like the kind of yeast you use to make bread with, they evolved to work anaerobically. So they will work anaerobically first and foremost, and then revert to aerobic respiration if they need to. So both of these are pretty much just glycolysis, really. It occurs where glycolysis occurs, in the cytoplasm, but they have different end products. Okay. So in mammals, pretty much glycolysis happens, but when you get to pyruvate being made, it's converted to lactate instead of acetate or an acetyl group. So it's got lactate instead. Lactate's quite harmful, it's acidic, it lowers the pH of your blood, it can build up in your muscles, feels very painful. So when you're working anaerobically, you end up with quite a lot of pain. And at the end, this lactate can be taken up to the liver and reconverted back to glucose, which is gluconeogenesis. We're still able to produce a little bit of ATP through substrate level phosphorylation, and it's all kept alive by this coenzyme NAD going back and forward, moving hydrogen molecules around. Pretty much the same in yeast and plants, except the pyruvate's converted to ethanal, which is a 2-carbon molecule, and then to ethanol. It's got a waste product of carbon dioxide.